Episode 1 described virtual screening as the stage that cuts wet lab hit finding from months to days, and Episode 8 contrasted it with generative design. This episode is the deep dive on screening itself, the workhorse of the field. It's less glamorous than generation, but it's deployed at enormous scale across real pharma today. So what is screening, precisely? You take a library of candidate molecules, and you computationally score each one for how likely it is to bind a given target, so that you only ever physically test the top-ranked few. That's the whole idea, ranking before synthesizing. Instead of running an expensive lab assay on every compound, you run it only on the handful the model is most confident about. Everything else in this episode is about doing that ranking well, and doing it at enormous scale. The classic method is molecular docking. Docking predicts how a molecule fits into a target's binding pocket, and scores the quality of that fit as a proxy for binding strength. Schrödinger's glide is the canonical docking engine, and, as we noted in episode 2, Schrödinger's platform is reportedly used by all of the top 20 pharma companies. It's the unglamorous, already at-scale counterpart to the flashier generative headlines. Docking actually predates the whole deep learning wave. What's new is that machine learning is now layered on top of it, to make the scoring both faster and more accurate. Here's the modern problem. Screenable chemical libraries have exploded to billions of make-on-demand compounds. Docking all of them exhaustively is computationally brutal, you simply can't afford it. There are two responses. The first is machine-learned scoring functions, replace or augment the slow, physics-based scoring with a learned model that approximates it far faster, so you can rank many more compounds per unit of compute. The second, and this is the clever one, is active learning. You dock just a small sample, train a cheap model to predict the docking scores, use that model to skip the vast majority of unpromising compounds, and then only dock the slice it flags as promising. That's what turns a billion-compound screen into something actually tractable, and it's the standard way ultra-large libraries get searched. So when do you use screening versus generation? Screening is grounded in real, orderable compounds, which means there's lower risk that your winner turns out to be something no one can synthesize, but it's bounded by what already exists. Generation, from episode 8, can invent genuinely better molecules, but it may hand you ones that are hard or even impossible to make. In practice, they aren't rivals at all, they're combined. Generative models expand the space of possibilities, screening-style scoring triages that space down, and both of them ultimately feed the very same wet lab validation bottleneck from episode 5. One is the expansion, the other is the filter. And the honest caveat. Docking scores are approximate. A good docking score is a hypothesis, not a guarantee, and false positives are common. The reliability of these machine-learned scoring functions on genuinely novel chemistry is, once again, the episode 5 generalization question in a new costume. So hold the right expectation, screening narrows the haystack dramatically, from billions down to hundreds, but it does not remove the need for the bench. It just makes very sure that the expensive bench work is only ever spent on the compounds most likely to actually work.